So, good morning, everyone. I hope you're all doing fine. Welcome to yet another lecture of molecular biology, the 3 days workshop. Please confirm if the voice is clear and there is no lagging in the video. Yeah. If my voice is fine and you can see my video clearly. Is my voice coming to all of you students? And the picture, is it fine so far? You can see my whole screen clearly without any errors. Right, 720p. That's good. All good, all good. So, yesterday we were discussing about basically yours basics of molecular biology, right? So, till where we did last time? Just a second. Till where we did, students, yesterday? And I can't see the video of for today actually, for yesterday on the internet. It's somehow deleted, unfortunately. We did till translation, okay. Yeah, we did this translation process. DNA replication was done, transcription was done, translation was done very nicely. Yeah, yeah, yeah. Good. Yeah. So let's start from this part, the genetic code. I hope you're all fresh, had a good breakfast of the champions, an energetic coffee, and yeah, with a fresh mind. And no one is lying in the bed in just a lazy position, all in the chairs with fully ready to have more knowledge about the um, basics of your molecular biology. So here we will talk about now genetic codes. In order to make your genetic codes, they are basically made up of 3 amino acids. Yeah, these three not amino acids, 3 nucleotide base pairs. That is AC, basically A and UCAG. So on the left hand side, you write UCAG. On the top, you write UCAG. On the right, in the each box, you write UCAG 4 times. So you will get 16 boxes. Then start writing 4 names in each box with taking 3 letters from each side. So UUC, UUA, UUG, right? So in this way, you have one box completed. Then you have your serine, you have leucine, you have proline, and so on. So your start codons will be your methionine, and your stop codons will be UAA and UAG. And so on. So now the next part is your genome. What is genome? Actually, genome consists of all the genes of human or any animal of which anyone you are looking for, any specific. So they belongs to that part. So normally the 98% of our human genome is not coding for any protein. It's still unknown what is it used for. But merely 2% of our human genome is known for coding proteins. It's actually helping to produce proteins. The rest, it's of, I don't know what's the use. Still scientists couldn't find it. So actually, genome is an entirety of an organism, hereditary information. It is encoded either in DNA or many types of viruses in RNA. The genome includes both genes and non-coding sequences of the DNA. So then within the genome, there is a field called comparative genomics. It's a very hot, I would say, topic if somebody is interested with the genomics part to study about genomics. It's one of the hot topics, that is comparative genomics, in which you compare each genome, your each gene, with the another species in order to check the evolutionary connection between two different species. So here in this table, we have established a very nice establishment of various species from human sapiens, mouse, Drosophila, Arabidopsis, C. elegans, Saccharomyces cerevisiae, Escherichia, Haemophilus influenza. Yeah. So all these different species. So you can check first maybe the chromosome numbers. So human and mouse, they have around 46 chromosomes, and your yeast has around 32 chromosomes. So they are mostly, if you want to study with human diagnostic diseases, so these two organisms are highly used for that. So these two organisms are highly used for the same. But if you want to study about genetics, yeah, so we can see the size base pairs of genes is 3.2 billion, here 2.6 billion in a mouse, and in Drosophila it's 137 million, yeah, and in roundworm it's 97 million. So, and this is the least, also the 12.6 million. So these are for the genetic studies, so these are also very nice models. And for the plant, if you want to study any traits or transgenic crops, so this is 100 million with 10 chromosomes. And for the prokaryotes, that's a separate field. It has only one chromosome, around 4.6 and 1.8 million size pairs of base pairs of your genes in E. coli and Haemophilus influenza. So that's how these comparative geneticists, what they do is they take the whole genome and they start comparing of each gene, and it took actually maybe 2 PhDs or one postdoc in order to complete this part to study this. So why genome analysis is necessary? It's basically it's a prediction of genes in uncharacterized genomic sequences. We are able to obtain the complete sequences, as many as genomes as possible, for genetic modification and genetic modifications to develop new varieties at a faster rate, like to produce your transgenic crops, GMOs, BT cotton, BT brinjal, and so on. So we are done with the basics of molecular biology so far. That is the core subject of molecular biology. Now I will take you some tools, some aspects of molecular biology which are very helpful in the industry, from the industrial perspective, from the job perspective. That is most likely you will get the question asked that what are the techniques that you know related to molecular biology, what is the concept behind them, and what do you know. So this is a 3 days workshop, so I cannot go in deep in 3 days, so today, but I will give you little bit touch towards each other. So what is not, so I will give you for sure touch for each other. So for gel electrophoresis, so the basic principle is that the